This video demonstrates the different steps in assay for neural induction in chick embryo. First, the host embryo is explanted in new culture. The donor embryo is explanted in saline and Henson's node is labeled with fluorescent dye. Henson's node is excised from the donor embryo and transplanted to the host embryo. The host embryo is cultured for 24 hours, fixed, and processed for photooxidation under fluorescence with DAB. The embryo is processed for in situ hybridization with a neural marker.
Hi, I'm Delphine from the laboratory of Rick Finnell at the Center for Environmental and Genomic Medicine at Texas A&M University in Houston. Today we will show you a procedure for performing neural induction assays in the chick embryo. We use this procedure in the lab to study the effects of antiepileptic drugs on the neural inducing properties of Henson's node. So let's get started.
For the neural induction assay, eggs need to be prepared. These eggs should be incubated laying on their side in a humid incubator for 13 hours to Hamburger and Hamilton stage 3+, or HH3+. Next, prepare the host embryo using the new culture method, which has been demonstrated previously in Jove. Cover the surface of the host embryo with 200 microliter saline. This will facilitate later transfer and positioning of the donor graft. Finally, cover the assembly with an inverted Falcon 35mm culture dish. To explant the donor embryo, open the egg by tapping on the shell with forceps. Remove top of shell and discard. Remove the thick albumin with forceps, and tilt the yolk sac with coarse forceps so that the embryo faces upwards. After removing the albumin, use fine scissors to cut a square of yolk sac around the embryo. Remove the embryo from the yolk with a spoon and place in a dish containing PBS. Next, use forceps to detach the donor embryo from yolk and area pellucida. Once it has been detached, transfer donor embryo to a Silgard covered dish containing PBS. To label and transplant the graft, use a microelectrode puller to pull 50 microliter glass microcapillary pipettes. Under the microscope, cut the tip of the micropipette using fine forceps. Place micropipette in a petri dish lined with a ribbon of modeling clay. Next, prepare the dye solution, which will be used to label the graft prior to transplantation. First, prepare a stock of dye eye in 1 milliliter absolute ethanol at 0.5%. This stock can be stored at -20°C in the dark. Then, in a heat block set at 45°C, mix pre-warmed 10 microliter dye eye stock to 90 microliters 0.3 molar sucrose. This temperature is necessary in order to prevent precipitation of dye eye and formation of insoluble crystals. Briefly spin the solution on a mini spectrifuge for 5 to 10 seconds. The dye eye solution is now ready to use. Using insect pins, secure the donor embryo on the bottom of the Silgard covered dish, dorsal side up. Once the embryo is secured, backfill the micropipette with dye eye. By applying gentle pressure using an aspirator tube assembly, apply a small bolus of dye eye to Henson's node. Repeat. Make sure the entire Henson's node is labeled. Next, using a microcapillary pipette or a microdissecting knife, cut Henson's node. The dorsal side of the node can be distinguished from the ventral side because it forms a groove, whereas the ventral side of the node is flat. If necessary, mark the dorsal side of the graft with carmine powder. This will ensure that the graft is positioned with proper dorsoventral orientation. Using a 10 microliter Gilson pipette, transfer the graft to the host embryo. Using a microcapillary pipette, bring the graft close to the area pellucida, area opaca boundary region at the level of Henson's node of host embryo. Next, use a microcapillary pipette or a microdissecting knife to make a small incision, 80 to 100 micrometers, in the area pellucida, area opaca boundary region at the level of Henson's node of host embryo. Using a microcapillary pipette or a microdissecting knife, position the graft so that the ventral side faces towards you and the posterior end is parallel to equivalent region in host embryo. Once in position, remove saline from host embryo, making sure the graft remains positioned in close proximity with host site, and process the host embryo for new culture for 18 to 22 hours as described in our previous submission. After 24 hours of culture, the embryo is removed from the incubator and pinned down in a Silgard dish containing PBS. PBS is removed and replaced with 4% paraformaldehyde. The embryo is then fixed for 24 hours. Dispose of the fixative in a suitable hazardous waste receptacle and add PBS to the fixation dish. Remove insect pins with forceps. After the pins have been removed, use a blunt end pasture pipette to transfer the embryo to a scintillation vial containing PBS. Wash the embryo twice in PBS for 5 minutes each and once in tris buffer for 5 minutes. After washing, transfer the embryo to a glass cavity slide containing 0.1 milliliters tris buffer. Replace the tris buffer solution with 0.1 milliliter DAB solution. Dispose of Eppendorf tip in bucket containing a bleach solution in order to decontaminate DAB. Place a cover slip on top of cavity slide. Under FETC, fluorescein isothiocyanate fluorescence, focus the objective of the microscope on the graft. Donor cells will appear fluorescent red, and expose to FETC fluorescence until all fluorescent red cells have become brown. This happens as a result of the difluorochrome being photoconverted to insoluble brown crystals by exposure to FETC excitation wavelength 488 nanometers in the presence of DAB. This process takes between 20 minutes and 2 hours. Following completion of the photooxidation reaction, remove the cover slip using fine forceps. Transfer embryo to a 20 milliliter scintillation vial containing PBTW, or PBS with 0.1% tween 20. Finally, deactivate DAB from cover slip and glass cavity slide by incubating in bleach solution. To process the embryo for visualization, proceed with in situ hybridization, incubating the embryo for only DIG labeled probe. Next, begin photographing and sectioning as described in Joe Video 2. Here is an example of a neural induction assay. Initially, the graft is positioned in the border between the area pellucida and the area opaca of the host embryo. Both donor and host embryos are stage 3+. After 9 hours in culture, the graft starts to differentiate and induce what appears as a miniature stage 8- head. After 24 hours in culture, the neural induction is complete. Following fixation, the embryo is processed for photooxidation. Initially, donor-derived dye-eye labeled cells are shown to have proliferated to form a miniature notochord as seen here in red. After photooxidation, these cells appear brown. The embryo is then processed with whole mount in situ hybridization for the neural marker OTX2, developed in the laboratory of Dr. Boncinelli. The donor graft is shown to induce the expression of OTX2 from host-derived tissue.
We've just shown you how to perform a neural induction assay in the chick embryo. When doing this procedure, it's important to remember to remove all fluid in the area surrounding the graft following transplantation using a pipette. This will ensure that the graft is securely positioned and that it will not move away from the host embryo during the culture. So that's it. Thanks for watching and good luck with your experiments.